Welcome to the cutting edge of cardiovascular sciences. I'm your host, John Cooke. I'm professor and chair of the Department of Cardiovascular Sciences here at Houston Methodist. And today we're going to be talking about single-cell omics and how single-cell omics will change cardiovascular sciences. We are fortunate today to be joined by two experts in the field, Dr. Ruli Gao, who's an assistant professor here at Houston Methodist Research Institute, who has developed some of the technology for single-cell omics and has done some wonderful work that we're going to be hearing about in a moment. And joining us from Stanford University is Dr. Sean Wu. Welcome, Sean. Sean is associate professor at Stanford University School of Medicine in the Division of Cardiovascular Medicine, and also associate director of the Stanford Cardiovascular Institute. It's great to have you on the show, Sean.
Great to be here. Thanks a lot.
Well, I think what we'll do, we have someone who is an expert in the technology, who understands how single-cell omics is done, how it's performed, how we get the data. And we have Sean Wu also, who's going to be telling us about the applications. So let's begin by talking to Dr. Ruli Gao about the technology of single-cell omics. What is single-cell omics, and what is its power to tell us about single cells and give us new insights into disease?
Yeah, glad to that. Dr. Cooke, thank you for having me. So I'm very glad to share some single-cell sequencing technology and also some computational method to everyone. So we have been focusing on developing both computational method and also single-cell sequencing technology for analyzing single-cell genomics. So today I just give a very short overview of everything. So it's going to be very superficial. So I'm going to share my screen over here. So this one just show you a very short part of the whole list of single-cell sequencing technology. So nowadays, single-cell sequencing technology are really emerging as powerful tools for analyzing the heterogeneous cell types and their transcription programs inside the human body or any human organs or mouse organs or whatever, any organisms. So the single-cell technologies are available for analyzing single-cell transcriptomes, single-cell genomes, and also single-cell epigenomes. Here I also list the spatial omics, which is really superb. So I really like this technology. So after this, I just want to quickly go over some technologies that we have been developing and some other popular technology other people are using as well. So this one show you one of the single-cell DNA sequencing technology that is developed based on the first single-cell DNA sequencing method that's developed for human mammalian cells. So this method starts from single-cell isolation, followed by library preparation, and last step is sequencing with next-generation sequencing technology. So here I show you, usually single-cell sequencing starts from tissue dissociation to make the single-cell suspension or single nuclei suspension. Then we flow sorted these single cells or single nuclei into individual wells in 96 well plate. As such, so each well in this plate will have only one cell or only one single nucleus. Then we perform the sequencing library preparation for each individual cell. So during this library preparation step, we introduced cell barcodes. So each cell will have a unique cell barcodes, nucleotide cell barcodes. So after library preparation, we poured all of the cells together, treat them as only one sample, and send that for next generation sequencing. So this method was based on Dr. Nick Nevin's first single-cell DNA sequencing method. So the first method sequenced one cell at that time, one cell in one lane, so that's quite expensive, around $2,000 per cell. So with this multiplexing, we were able to sequence around 100 cells per lane. So the cost dropped dramatically by 100 times. So, but nowadays, that's in 2016. Now we can sequence more, like around 400 cells. So the cost dropped to around $5 per cell. So this is for a single cell whole genome sequencing technology. So next, let me go over this quite popular droplet system. This is widely used for single-cell transcriptome analysis. So this method is based on a droplet system. So this method uses the droplet emulsion approach. So during this emulsion approach, it wraps individual cells and chemicals and also barcode gel beads into one droplet. So within that one droplet, reverse transcription reaction happens to make the individual cDNA, then library preparation followed by this step. So in each droplet, the cell barcodes were introduced. So after this library preparation step, you can pull everything together, but differently, each cell is barcoded. So after sequencing, you can, based on the barcodes, you can really tell apart individual cells. So this technology is very powerful and really high throughput. So with this text genomics Chromium system, you can analyze up to 10,000 single-cell transcriptome in parallel. And they further continue develop some other layers of technology. So now you can sequence 3' MIs, 5' MIs, and you can also sequence a TAC-seq and VDJ or copy numbers with their platforms. So it's quite amazing. Here I introduce another method we developed previously, called nanowell single nuclear RNA sequencing. So this method is different from droplet, that's liquid. So this one is focusing on developing technology for isolating single cells. So we developed together with the Wafergen. Now it's bought by Takara iCell 8 system. So it's a nano dispenser system. So for this one, we again started with single cell or single nuclei suspension. Then we can dispense this cells into a nanogrid nanowells. So then this system also attached with an automatic nanowell imaging scanning system. So with this system, you can check individual nanowells to see if this well is empty or has single cell or has doublet or a triplet. So this dispension follow Poisson distribution. So on average, you have around 30% of the nanowell will have single cells. So the advantage of this method comparing to droplet is that you can actually use the image system to check the morphology of the cells that you isolated. So the throughput is quite okay. So we can analyze 500 to 2,000 single cells in parallel. So with this system, we developed the single nuclear RNA sequencing method that was first for analyzing human cancer cells. So, and we also compare the whole cell transcriptome and the nuclear transcriptome. So with this method, we show that the nuclei fractions of pre-MIs and the whole cell MIs, their expression levels are highly concordant, can achieve up to 90% of the total transcriptomes, except for some genes like nuclear functioning genes, transmemory genes, or pseudogenes. So those are differentially expressed between nuclear fractions and cytoplasmic fractions. So it's kind of expected, and this is a very powerful tool. So here I just briefly show you some, just demo the applications of single cell sequencing onto cardiovascular and heart disease. So here in this one study that's published in Natural Cell Biology by Dr. Wang in whose group. So in this paper, they analyze single cells from the heart. They try to analyze the total cellular compositions in heart, and they also try to analyze different cell populations in different compartments. So here just show you the isolated single cells from left ventricle size and left atrial. Different areas. We try to analyze the cells from different areas, and they also apply different single cell isolation method, and specifically they developed a method try to enrich cardiomyocytes, because these cells usually die very quickly without the nutrition oxygen enriched environment. So they isolate cells from different areas and send that. Here they applied actually nanoweld the single cell sequencing method. They check individual cells, so they only analyze the live cells. So after sequencing, they performed unbiased clustering and defined several clusters. So they first define cluster, then they annotate the cluster based on your knowledge of the cell type specific markers. So they define one cluster of cardiomyocytes, one cluster of endothelial cells, a big cluster of fibroblasts, a big cluster of macrophage, and one big smooth muscle cell clusters. So after this unbiased detection, they further check the differential expressed genes between different cell types. So it's really a unbiased approach to analyze these cell types and their translational programs. So further, they have a focus on the cardiomyocytes. Then they check the different gene transcription programs in different areas in the heart. So they specifically analyze the contraction genes. Then they show the difference of these contraction gene expressions. They are expressed in cardiomyocytes, but they are different. So here they observe a set of contraction genes specifically expressed in LA region, and some set of genes only specific expressed in LV genes, and some LV regions, and some are shared into different regions. So similarly, they analyze the metabolic gene sets, so show some difference, and also the secretion membrane genes are also differentially expressed between different regions. So they find both inter and intra-compartment difference. So actually, you can further recluster each cell types to define the subcluster of each cell type. So they further divide for fibroblasts, keep on dividing the fibroblasts into several subclusters. They do similar things for endothelial cells, just keep on reclustering and develop a new subcluster. So based on, so you have many subclusters, and based on you can study the cell-to-cell interactions based on the receptors and their ligands. So how do you do that? This we call interactome. So basically, you check the expressions of receptors in one cell types, then you check the concordant expressions of their ligands in another cell types. If they have high concordance, you can infer that they might have kind of association or interactions. So this is another application, and another one they analyze some heart failure recovery patients. You can use the transcriptome to check the different cell types changes during the recovering process. Okay, so here they show that recovered patients have populations of cells that express similar patterns of the endothelial markers comparing to the normal cells. but in the patient that failed the recovery, they didn't detect this kind of rescue of the endothelial gene expression. So this shows that some recovery of endothelial cells during this heart recovery process, heart failure recovery process. This based on the single-cell transcriptome analysis. So next, I want to talk about the delineation of the cell fate conversation. So with single-cell RNA sequencing, you can detect all kinds of single-cell subtypes, and also you can check the cell fates. Say you say these cells are endothelial, these cells are mesenchymal or fibroblasts. So you really don't know the relationship between these cells. But with the transcriptome, of course, you can infer the pseudotime. So we call pseudotime. So based on their transcriptional programs, so we infer the relationship between the cells. So it's really not a tree. We call that pseudotime. Yeah, potentially we will see for instance, in a heart recovery, we detect a subset of endothelial cells, and we also observe decrease of fibroblasts. So potentially there's some relationship, right? So, but we really don't know. So another type of study we call cell lineages. So it's not, it's quite different from the original microscopic observation of cell lineages. So with single cell genomics, we can use DNA mutations as markers to track the relationship between cells. See if you use stable genetic DNA mutation markers. So this marker is supposed to be stable between different generations. So it should be stably propagated between generations. So, so single-cell DNA sequencing can be reliably used to track cell lineages. Then the single-cell transcriptomes can be used to infer the pseudotimes. So there's cell fate tracing. So ideally, if we can combine these two approaches together, we will see how the cell fate will change, or how the cell will differentiate during the cell division steps. So ideally, we have two layers of study merged together. So this leads to the single-cell multi-omics. So this kind of technology is focused around the isolation of different layers of omics from same cells. And this technology is centered around transcriptomics. Like you can combine transcriptomes with genomes. You can combine transcriptome with DNA methylation or chromatin accessibility, and transcriptome with proteome. So just a combo. So our lab was focusing, developed a method, a computational method called copycat. We inferred the genotypes from single-cell transcriptomes. So really, after this inference, for each individual cells, we will have transcriptome information. We will also have genome-wide copy number information. So we really can, using transcriptome data, to dissect the subclonal information within tissue. Mostly this is developed for tumor tissue, but potentially we can apply to cardiovascular using other genetic markers, like let's say mitochondrial DNA mutations or tandem repeats, something. So this is a demo. So with that, I somehow it didn't show up here. I want to finish my presentation with this single-cell computational challenges. So there's tons of challenges in analyzing this single-cell data, multi-layers, high throughput. So it's very powerful, but also very challenging to analyze this data. So thank you for your attention.
Thank you, Ruli,
Yeah.
for introducing to us this powerful technology of single-cell omics, some of which technologies you've had a hand in developing. I think, for me, one of the most important things that I learned from your talk was that we've got these technologies now that can give us so much information in a single cell and really start to finely delineate different cell populations. When I was in medical school, it was a long time ago, when I was in medical school, we were told that there are 225 different cell types.
Yes.
That was clearly not correct. There's a lot more cells than we knew exist. You showed us this paper that was just published in Nature Cell Biology, where 4 different types of cardiac fibroblasts were observed in that study. I think 4 or 5 different types of endothelial cells within a heart.
Right.
We always knew that the left heart and the right heart were different somehow.
Yeah.
You know, now we're beginning to understand these different subpopulations that may be contributing to the differences between the left heart and the right heart.
Yeah, exactly. So, just single cell sequencing for heart tissue. But you know, the Human Cell Atlas project, they try to sequence every cell states of the whole human body. So it really identify a lot more cell types and also cell states comparing to the original assumption, like 200 cell types in the human body.
Yeah.
So it's really amazing.
We're going to learn so much about disease as you apply these technologies, and I know you're doing some of that here at Houston Methodist, applying these technologies to disease processes, and I think we'll learn about cells that play a role in disease. We'll learn about cells that play a role in recovery from disease.
Absolutely, yes. So so the power of tools, so it should have amazing applications, right? So we put lots of effort in developing this technology, developing the computational tools. So we want it to have amazing applications. So here at Houston Methodist, we really have rich resources for analyzing different clinical resources. Like we have heart failure samples and heart transplant samples, and are different kind of vascular disease from real human body, right? They're not mouse model. It's really amazing. So I believe the application of single cell omics to this human clinical samples will lead to great discovery in the field.
Thanks, Ruli. Well, let's move on to Sean. Before we move on to your talk, Sean, did you have any questions for Ruli or any comments on what you just heard?
Yeah, so it was beautiful talk, really. I really enjoyed the aspects that you cover, overview from the genomic into the transcriptomic, and a little highlights on the proteomic and metabolomics as well. I think what you mentioned about the challenges of integrating all these data to be able to extract the information that are going to be helpful for us to understand biology and move forward with drug discovery is really just a very important direction, in terms of how we're going to be thinking about, the way to do science, in the future. It may be a little daunting, I think, for people who have been more traditional in thinking about how to do biology, but, I think this is, part of what we do as scientists is to learn new things. So definitely very much looking forward to see all the new developments that coming out from your research group on these computational tools.
And you mentioned, Sean, the clinical relevance of this. I mean, there's obviously scientific merit in understanding these different subpopulations and their role in pathobiology of disease. These are going to provide clues. These insights will provide clues for new therapeutic avenues. So I'm glad you brought that up, because that I think is an important point for the clinical relevance of what you're doing, you and Dr. Gao are doing. Well, why don't we turn now to hear what Sean Wu has to say about the application of this powerful technology to cardiovascular disease. Sean.
All right, sure, great. Thank you so much, John, and thanks for having me join this. Actually, hang on, sorry. Let me just switch, share screen. Okay, so hope everybody can see the slides that I'm going to be presenting. So yeah, again, John, thank you so much for having me here at the Cutting Edge Cardiovascular Science Seminar. And I'm going to be talking about how the way these single cell approaches are helping us understand and discover new biology. And I think, certainly for some of the audience who are a little bit remote from doing the science themselves, I'll start pretty, easy. So here certainly is something everybody's very familiar with, and that is what we call the central paradigm in biology. You have what comes from the DNA, which makes the RNA, and to protein. And we have always sort of thought this rather linear process of going, from the coding into the transcript, into the protein. But I sort of think of this as something that we've been thinking about for many, many decades, if not longer. And to try to understand genes that are relevant in disease, it's a little bit, sort of like, here you have this gentleman here sitting here fishing, and as this gentleman was someone who was interested in, say, the lipid biology and trying to understand how the lipids are regulated. And I think for most cardiologists, if not all, the regulation of lipids has certainly it's something that has been front and center of what we do as cardiologists. But as you can see, the process of trying to find these new genes and discover ones that are important for disease, doing it this way, one at a time, is extremely slow and very inefficient. So what has it that's been driving this interest in wanting to discover more genes? Well, so one thing I think it's important to think about is the costs and the challenges of discovering new drugs. And I think most of you probably have heard of what people have called Moore's law, and that is the pace of which the processor speed and the storage data has been doubling roughly every 18 months. And then so what people do in drug discovery sort of rewrote this, called Eroom's law, which is the reverse of Moore, which is this is the cost that it would take for the number of drugs that it takes to be created for the cost. So here you can see that I put this big arrow here, a red arrow right at about year 2000, and that's where we have crossed the line that it takes about $1 billion to make one drug. And then as this line keeps going further and further down, by the time we get hit about now, we're currently at about $2 billion to make a drug. So on this curve, it essentially represents a $1 billion gives you half of a drug. So the process is definitely becoming less and less efficient over time, and it takes longer and longer to develop new drugs on the order of 12, 16, or even longer to develop one new drug. So what this means is that we really need to pick up both the pace and also figure out a way to reduce costs. And then that one reason for why these omic technologies have come along is to try to figure out how to find the most important candidate genes that regulates biology in a way that's much more efficient and higher throughput. And then so along with this idea of trying to be better and more efficient is the idea of needing to be more precise. And I think all of us have known back 5 years ago, former President Obama started the Precision Medicine Initiative. And the idea for Precision Medicine Initiative was said to be, delivering the right treatment at the right time and every time to the right person. I think concept certainly sounds great. And I think most of us as clinicians have been already doing things as precise as we possibly can. And I think what has become the new change, the game changer, is the ability to sequence DNA. And then so I think the idea is that if we have better ways to sequence to understand the genome, and then also subsequent to that, the different products of the genome, then we also could identify the various differences from individual to individual to provide that kind of greater granularity and resolution to the understanding of biology. So I think that, given that these have been some of the main interests in understanding the biology on a faster level, everyone now is essentially turning into looking at not just the single DNA, RNA, and protein, but actually looking at it at the entire genomic, transcriptomic, and proteomic level. And I think Ruli has just mentioned very well some of the new technology that have come along in doing things at this scale. And so this would be essentially like, instead of just one person fishing for one interesting gene, you now have the entire fish trawler that scoops up as many genes to look at the same time. And then hopefully that will speed up the catch and also the time that it would take to find interesting new genes in biology. So that's sort of the impetus of what people have in terms of moving into this sort of faster and faster pace, and also greater and greater data. And I think Ruli has also mentioned about the different level of the biology, from the genome to the epigenome, which are the regulation of various different parts in the genome not coming in the coding sequence level, but also transcriptome, which we'll talk furthermore in a little bit, along with proteome and metabolome. And in terms of the technologies, I think the genome, epigenome, transcriptome certainly has benefited greatly by having tools such as the next-gen sequencers that have been developed to do high throughput sequencing. But in terms of the proteome and the metabolome, these also have benefited by the availability of mass spectrometers that is allowing people to be able to generate lots and lots of new information at the protein and the metabolite level. And I think one further challenge that it sort of generates is all this information at the genome, the transcriptome, the proteome, and the metabolome level really requires ways to understand how to integrate all of these information and data into an interacting network to be able to identify what some of the key nodes that are important in regulating the biological process. And I think things that Ruli mentioned about developing the computational methods to understand the way these different genes and also the transcripts and the proteins interact in real time is going to be extremely important. And so, I wanted to then focus beyond just looking at the specific technology for the genome. What is it then about the single cells? How do we get from trying to understand many genes and many products into wanting to understand them at the single cell level? So I sort of showed this slide because this is the biology that we worked on for more than a decade, which is understanding the development of cardiovascular cells. And so when we started out, the ways of doing science usually involve looking at cells in big populations. We identify early precursor gene for heart progenitors, such as gene like NKX2.5, that give rise to these different differentiated cardiac cells once the heart has developed fully. But we've also looked at some of the key regulators that have driven the process, such as a gene called yin-yang-1, which many people work on, including Bob Schwartz over there at Houston, and also many others. And we've shown recently that the YY1 prohibits the differentiation of these heart progenitors. And so while we were working at understanding the biology, what has been rather frustrating is that every time we try to see what are the potential for differentiation of these progenitor cells, we always get a heterogeneous population of end products that we're not always sure whether they represent true individual cell with very distinct phenotype, or we're just looking at a heterogeneous mix of cells with not truly distinct phenotype, but kind of a halfway one to the other. So this really prompted the interest in wanting to have a tool that could tell us that precisely what exactly is the identity of each individual cell that we are looking at, and also potentially find this variation within each population that we're looking at as well. So this is then how the transcriptomic for a single cell has come along, and Ruli has mentioned about lots of the different possible tools, including the ones from 10x Genomic Chromium, which is a droplet method, as well as Carole Bio, but the iCell that she described very nicely of the capturing of cells to look at the nuclear sea. There's certainly many other tools that have come along, but we have worked largely with the 10x Genomics Chromium system. And so one of the papers that we published a few years back was looking at understanding all the subpopulation of an embryonic cardiac heart. So in this case, we took the embryonic mouse from very early stages at day 8.5, 9.5, and 10.5, which roughly represents a transition between the first trimester within the first trimester and at the heart tube stages. And then so what we're able to do with the single cell transcriptomic is to show all the various different subpopulations within endothelial cell, the cardiomyocytes, to be able to break them down into even the subregions within this developing heart and be able to essentially use a set of markers that we identify for each region as a way to identify that cell. And then so this sort of gave us the impetus of being able to use the transcriptomic data to deconvolute the phenotype of each individual cells, which has really driven much of the research direction that we've taken on in recent years. But I wanted to also illustrate another use for single cell, which is using it to identify very small and rare populations. So this is a very nice study that was published from Aviv Rajiv and Jay Rajgopal over in Boston. And what they have found is that by doing profiling in the mouse lung and using single cell tools, they were able to identify this extremely small population shown here in the circle in this the Tisney plot. And this very small population turned out to be a small cell called the ionocytes, and that this ionocytes, of which otherwise would not necessarily be so interesting if it wasn't for the fact that this ionocyte found within the lung represent the only cells apparently that expresses CFTR. And as all of you clinicians know, that the CFTR represents the genes that are defective in cystic fibrosis. And what I think has not been previously appreciated is how that this particular gene that's causal for cystic fibrosis turned out not to be widely expressed in many different cell types within lung, but it's exquisitely in this very small population. So I think this really illustrates how having the single cell tool gives you the kinds of understanding that you would otherwise never been able to do if you did not have a way to know what cells are the ones that are exquisitely able to express certain genes. And then so somewhat related to identifying rare cell populations, we recently published a paper in the cardiac field in looking at the development of the cardiac conduction system. So I won't have time to talk about all the details of the work other than to just show you sort of much nicer, prettier picture version of what we found. Essentially, by doing a single cell transcriptomic profiling, we found new markers that represent markers specific for the developing conduction system in the mouse heart. And by finding these markers that are unique, we're able to immunostain and track all the different presence of these different conduction cells and create essentially a tree of this conduction system from the AV node at the top through the internodal tracts onto the SA node at the top through the internodal tract to the AV node, and then down through the His bundle and the Purkinje system. So I think this tool certainly has been very useful to try to understand some of the challenges that we've had in the past with cardiovascular biology. And then so a final example that I want to illustrate here is something that we collaborated with Steve Quake, who is a bioengineer at Stanford. And the project together with also Tony Weiss-Corey was to try to profile all the cells from all the different organs within a mouse heart. And in this case, this is a 3-month-old mouse heart where we dissected 20 different organs and harvested, in this case, more than 100,000 cells, which we analyzed 85,000 using both the 10x genomic system as well as the FACS sorting with the SmartSeq. And what we were able to demonstrate is what you see. Basically, this is kind of like in the entire mouse in 2D, all the different cells and all of the various different subpopulations that you can see are all displayed here. And since we contributed to the cardiac dissection and profiling, I just want to highlight the cells that represent the cell types that we're very much interested in, including cardiomyocytes and endothelial cells, smooth muscle cells, and fibroblasts within the heart. Subsequent studies have now taken on to further analyze. We recently published a paper together with my former postdoc, Guan Li, who is at University of Pittsburgh, showing that the subpopulation of endothelial cells are actually quite unique, including a population of lymphatic endothelial that are entirely transcriptionally distinct from the rest of endothelial cells. So lots and lots of work remains to try to understand these subpopulations, but we think the tool and the computational the data analysis capacity that will be coming forward would certainly help us understand more and more of these biologies. And so finally, I think I want to sort of leave you with a little bit of the historical perspective of transcriptional biology over the last 40 years. So for people who have been doing science in the 80s, I think to look at the different gene expression is using a tool like Northern Blot, which I sort of consider that as akin to riding a bicycle in the neighborhoods where you see, individual houses as you go by. It's certainly, a nice exercise, but it's very slow to get through to see too many things. So by the 90s, we started developing tools such as PCR and RT-PCR that now allows us to get a little more view at a time. So here it's like riding on the hot air balloon where you're looking down into lots more houses at a time and getting to see more landscape. And then as you sort of follow along the same logic, 2000s, we have microarray and RNA-seq. And then that is like riding on a jet plane where you now cover much more ground. And then in 2010, we started doing single cell RNA-seq. And the single cell RNA-seq now is like being on a satellite looking into the entire continent. And then so I guess the question is, what's next? And well, I think in the 2020s, we're essentially looking at the entire planet, and the entire planet requires new tools. And this is an example of the 10x Genomics products page. And, I certainly own no stock in the company, but I find that they are just quite innovative in creating new tools available for even regular investigator like myself, who does not necessarily create my own tool, to be able to do lots of different omics-based technologies that you can see, the transcriptomics as well as the genomics, and also looking at the various different epigenomic plus other variations upon that. So I think this definitely is a super exciting time, and lots of interesting biology will certainly come forth from all these different investigations. And with that, I want to just thank some of the folks. So I have the folks that are working on single cell. I highlight the work that I mentioned from Guan about the development, and Will with the work on the cardiac conduction system, along with Benny, a German medical student, and then some of the funding agency that supported our research work. And I want to thank all of you for your time for listening.
Thank you, Sean. That was a beautiful overview. Tell us a little bit about what you're thinking in terms of the application of this technology to the clinic. What ideas do you have in terms of applying this technology that you just talked about to clinical problems?
Yeah, so I think, where a lot of the progress has been made is understanding the inflammatory environment. And we know in cardiovascular field, inflammation certainly plays, roles in many different processes, including post-MI inflammation and also in the vascular wall inflammation. So lots of the investigators right now are looking at identifying the specific populations that are responsible for disease process. So for example, in the atherosclerotic lesion, it has been now looked at that there are unique population of smooth muscle cells that appears to be precursor that initiates and migrates and then eventually forms the fibrous cap. And to try to understand what are the cell precursor and the specific pathways that regulate them is going to be very important, I think, for trying to understand the issue of plaque stability. But for people interested in looking at MI, and I think there has been lots of work looking at macrophage subpopulations and how the macrophage is able to regulate the inflammatory and the fibrotic process post-MI, I think are going to be extremely interesting and unique in terms of being able to find new causal cells for certain processes, that we previously may not really appreciate and thought that inflammation was just some, sort of general processes, involving all the usual cast and characters. And I think the likelihood that they're actually very specific population, that are the key driver of the pathologic process that we're going to be able to discover by having tools like this.
Mm-hmm. Well, the Cardiovascular Institute at Stanford is lucky to have you, Sean. You're doing spectacular work, and appreciate that overview. If there's a little bit of time, and I'd like to just get your thoughts about some work that we've done recently here using single-cell omics, and I'd like to just get your thoughts about this paper that was published recently in Circulation. It's a bit controversial. What we have found essentially is that there's a subset of fibroblasts in the ischemic limb that participates in angiogenesis. So, you know, Sean, patients I see are patients with peripheral arterial disease, vascular disease. And so I've been studying peripheral arterial disease for many years, and we use a mouse model to mimic peripheral arterial disease. And what we did was to ask the question, are there fibroblasts that play a role in supporting the recovery from ischemia? And so let me go to the money slide right here. In the lower left corner is the result of our single-cell analysis of fibroblasts that we obtained from normal or ischemic limbs in mice. And these are fibroblasts as defined by a surface marker, FSP1. And the fibroblasts share a lot of common gene expression, like collagen A1. But what was interesting was that in the normal and ischemic limb, there's 8 subsets of fibroblasts as defined by this single-cell transcriptional profiling. And you can see in the heat map there clearly the columns running across that box representing different clusters of fibroblasts clustered by their transcriptional profile. And what we found was that there were 2 clusters that seemed to have angiogenic properties. Cluster 8 made angiogenic cytokines, and cluster 5 actually expressed some endothelial genes. And it's a little bit more difficult to see up in the upper right-hand corner. That's the change in the distribution of these clusters with ischemia. So in gray, the gray bars are the distribution of the cells in a normal limb, and in black is a distribution with ischemia. And you can see that clusters 5 and 8 increase dramatically during ischemia. And so looking at the transcriptional profile of these clusters 5 and 8, we were able to find surface markers that allowed us to pull them out and study them in vitro. And here's one slide from that work. And what you see in the first 2 panels are the cells cultured just in 2D. They're on a culture dish with collagen, chlorine collagen layer, and they look like fibroblasts. And I'm sorry this doesn't show up too well on the screen, but the fibroblasts at a 0 time point before ischemia were compared to those at 28 days in cluster 5 and 28 days cluster 8. And they look the same morphologically. When we put them into Matrigel, there were some distinct differences. The fibroblasts that we got pulled out at day 0 in the absence of ischemia look like fibroblasts growing on Matrigel. They form these clusters, little balls of cells. That was true also of cluster 8. Those are the fibroblasts that can make angiogenic cytokines. But the ones that in cluster 5 that could actually express endothelial genes in Matrigel began to look like endothelial cells and form these tubular networks, which is very similar to what endothelial cells do when they're in Matrigel. So the clusters that we discovered, these different subsets of fibroblasts, the 2 that had angiogenic properties, one of them actually seemed to be looking like endothelial cells under the right conditions. And we've done work showing that inflammation plays a role in cellular plasticity. Basically, inflammatory signaling opens up the chromatin to give the cells greater plasticity. and cells can basically reach into their genetic toolbox and pull out what they need to survive or adapt. We found that this recovery from ischemia was dependent upon inflammatory signaling, and what we did to show this in a number of different ways was we studied some animals that had one of the important signaling pathways knocked out, NF-kappa B. Up in the top left-hand corner in panel A there, we see matrigel plugs that we inserted subcutaneously into the mice that retained the NF-kappa B pathway. That's there on the upper panel. You pull them out a week after inserting the matrigel plugs, and they're vascularized. There's blood in them. Mouse vessels have grown in there. There's been angiogenesis. But in the animals where the inflammatory signaling pathway was knocked out, we saw a sparse vasculature. Similarly, at the lower corner in C, panel C, we looked at the same group of mice, their response to ligation of the femoral artery, so limb ischemia. And in red, the animals that had the inflammatory pathway knocked out, they didn't recover as well. And without going into too much greater detail, we believe that their poor recovery was in part due to this loss of inflammatory signaling that permits these clusters to change. So clusters 5 and 8, as I mentioned, were very important in angiogenesis, and we found in FACS analysis with lineage tracing that in the right lower corner there, that in normal animals we see a cluster of fibroblasts that actually, with ischemia, start to express endothelial markers, and those are absent in the animals in which we've knocked out the signaling. In this case, we've knocked out the inflammatory signaling with dexamethasone. So anyway, the point is that there are different clusters of fibroblasts within the limb, and I know this is controversial because in the cardiac arena, but in the limb it appears that there are fibroblasts that do support angiogenesis, and that one of these clusters seems to be able to transdifferentiate into something that serves the function of an endothelial cell. And we had some immunohistochemistry in that paper to support that idea also, that there were fibroblasts were transdifferentiating into endothelial cells. And so this is a new angiogenic process that we believe is important to study, and understanding the cells that are responsible for this angiogenic process, the fibroblasts that can participate in angiogenesis, might give us some new therapeutic avenues to treat ischemia, at least in peripheral arterial disease. So I just wanted to mention that and get your thoughts, and specifically with reference to the controversy in the cardiac literature, because I know that this topic of transdifferentiation of fibroblasts to endothelial cells is somewhat controversial in the cardiac literature.
Yeah, John, that was really beautiful work, and I remember from some time ago that you coined this very, very wonderful word called transflammation, and I think that probably lots of different people are continuing on, with that concept of, the inflammatory role in causing cells to transdifferentiate to a different phenotype. Well, so I think, in terms of what has been sort of looked at and what people are able to appreciate is that there are transdifferentiation that certainly happens. So we know very well that in, say, development or in disease setting, there is the conversion of endothelial cell through the endo-MT, mesenchymal transformation process, into a fibrotic, fibroblast-like. In fact, that, as you know, that is the way the valve have been developed during the early stages of heart development. So I think the cell type conversion is something that can and does happen probably more readily than sometimes we are necessarily, sort of willing to think of, because, once we sort of get a little fixated on certain process, that we tend to be, a little resistant, to change from what we know. I think the issue for looking at a conversion in the reverse direction of fibroblasts that then adopts a endothelial cell property in terms of things like gene expression, it certainly is possible for that to happen, and I think that probably hence raises some of the controversy when people observe expression of genes like that. I think one key issue, which I think we continue to struggle with, and I know you know this very well, is how to make the definition for what the different cell, that you would identify assigned to, because this has been something that many people who are interested in trying to do a direct programming, has had a lot of struggle with, which is introducing transcription factor and try to convert that into a specific cell type. And I think what over time people realize is that there are some phenotypic shift and transcriptional shift, but each step along the way into trying to be the actual equivalent to the in vivo derived cell, there's attrition in the process. So if you start from 100,000 cells, maybe by the time you get to the end, having the cells that truly represent the cells that you're comparing against the gold standard, you're now down to a much smaller fraction. And that process, attritioning, probably differs, from one cell type to another, and also with different processes. So I certainly think the inflammation is a very powerful process that could clearly drive the phenotype shift of the fibroblasts. But in terms of how well the fibroblasts have adopted the phenotype of a true endothelial cell, that I think is clearly where a lot of the controversies lie. But I think these technologies like single cell are really the ones that's going to help us figure out exactly where the cells are in that spectrum of the conversion, from one cell fate, to another. Lots of exciting things, I think. We'll be looking at in the future on this.
Yeah, thanks, Sean. And Ruli may have an answer for us with her genomic sequencing, right? The single cell genomic sequence combined with the transcriptional profiling might be able to allow us to do lineage tracing in humans, right?
Yeah.
Tell us about that.
Yeah, exactly. So if you're using single cell transcriptome alone, I would say like a single cell multi-omics is a solution. So if you're using single cell transcriptome alone, so to label the cell, the cell fit based on the non-cell fit markers, like say based on EpCAM, PECAM, you tell these cells are epithelial cells or endothelial cells. Based on certain genes, you annotate this cluster as certain cell types. But it's kind of plastic. The gene expression can change very fast. So I would say DNA markers, DNA changes that can be detected in the same cells, that would be ideal. So we will assume the DNA mutations, say, is more stable. So during this generation to next generation, it's very likely they will maintain the same mutations. So if you have certain mutation markers to track the cell lineages, then combine that with the cell fit, we'll have some answer about like the cell origins or source of new endothelial cells. So, but I think it's a very interesting story, transinflammation. So it might happen. So then you might have different source. So some cells come from transdifferentiation, but you also have some group come from proliferation, right? Proliferation. Potentially, you also have differentiation from the stem cells. So I think it's really important to dissect the origins of the, these cell subpopulations and try to compare the difference between them.
Yeah, that's a very good point. I guess it's what you're saying is that as a population of fibroblasts, they may have had a common progenitor where there was a somatic mutation, and then population of endothelial cells had obviously a different progenitor that may have had a different mutation, and so they have that embedded in their genome, those somatic mutations embedded in their genome, and then you can use that as a clue to where that cell came from. So if suddenly a fibroblast starts changing its colors and becomes an endothelial cell, it still has that genomic mutation from the progenitor from which it was derived. So it allows you to do some lineage tracing.
Right, exactly. So yeah, obviously in cardiovascular might have less mutation burden compared to tumor, but there's a big portion of intergenic region that's not well explored. Potentially we may have this kind of DNA markers to tell apart the lineages.
Yeah, very good. Well, we need to continue this discussion amongst the three of us and collaborate. I mean, this is fascinating. And Sean, I really do appreciate you joining us for our program today. And Ruli, thank you for joining me in the studio. And I think we're going to call it quits right here, but we have lots of more work to do. Thank you, Sean. Thanks for joining.
Right, great. Thank you so much. Thanks for having me.
Thank you.
Absolutely. And thank you everyone for joining us at the cutting edge of cardiovascular sciences.